Okay, yeah, thanks for the warm introduction. Yes, yeah, my name is Garrett Roell. I'm from the MBBE. For those of you who don't know, it's Molecular Bioscience and Bioengineering in CTAR. Today I'm going to be talking about metabolic models and machine learning for bioprocess optimization. So I want to begin by talking about some motivations. So historically, we've used fossil resources like petroleum fed to refineries to make fuels, but also chemicals. That can be used for clothing, like synthetic clothing, or for fragrances, or for pharmaceuticals. So it's a wide range of products that come from these fossil resources. And going forward, we want to replace that because they're not renewable, and I guess the price can fluctuate a decent amount, as we've seen the recent history. So starting in the 1970s, corn and sugarcane were fed to Saccharomyces yeast to make ethanol biofuel. And this was good, but two key shortcomings were using food to make fuel is not the best because it increases the cost of food. And also ethanol is not the best fuel source because if you have too much ethanol in your fuel, then it attracts water that can damage engines. So going forward, we want to use less valuable resources like waste resources like fat, oil, and grease. But we'll need to have special microbes, like non-model microbes, in order to make high-value products. So things like nutritional supplements like beta carotene and astaxanthin, or sustainable aviation fuel, like this dimethyl cyclooctane. And I'm going to be focusing on non-model microbes. So well, the reason to do that is because even though model species are great, we understand their metabolism well, and very many genetic tools exist to modify them. Uh, they don't consume the sort of carbon sources that we want natively or produce the products that we want natively. So we would need to engineer them extensively, and that leads to problems. So we want to use non-model species like Yerwaria lipolytica or Clostridium carboxyliborans because they have those properties, but unfortunately, we don't understand their metabolism very well yet. So that's what we want to do going forward. So I'm focused on fermentation, and the most interesting thing that we want, or the thing that we're trying to optimize the most, is titer or final product concentration. But there's many different inputs that go into this. So things like the bioprocess conditions, like the pH and temperature, and carbon source, and also the strain genetics. So like which heterologous genes do you add, which genes do you knock out, and how do those affect the metabolic burden? So when you add more enzymes, then it takes up the resources ATP and ADH and I guess amino acids. So we want to try to figure out what is the best way to optimize the system. And the way that we want to do that is using the design, build, test, learn cycle. So it applies broadly to engineering or science concepts, but for synthetic biology and fermentation, we're interested in like designing the strains, so choosing the genes to knock out or overexpress or add, actually making the strain by doing the genetic engineering, like adding plasmids or modifying the chromosome, actually doing the fermentation and then trying to learn from it. And my work is focused on modeling. So was interested in the learn and design phases. So what approaches are we going to take? So we're going to do integrative omics analysis. So we're going to look at genomics, like which genes are present, transcriptomics, like which genes are expressed the most or the least, and how does that vary based on the carbon source. Proteomics, which proteins are most present, and then metabolomics, how do those enzymes affect the concentration of the different small molecules. Also going to be doing mechanistic modeling. So the keyword there is mechanism. So we're focused on trying to map all of the different reactions. And when you put all the reactions in a network, that sort of sums to the metabolism of the cell. And mathematically, the network can be represented as a matrix shown up there. And then we can say, like, try to figure out what is the potential of the cell, like some things like growth rates or maximum product yields. And these first two methods require extensive like domain knowledge. So we also want to try to see if we can use data-driven methods like using machine learning and AI to connect the complex inputs to outputs in ways that might not be possible with the first two methods. Okay, so the first sort of story I want to tell is about Yerwaria lipolytica multiomics. And the goal of this project is to convert waste oils, and this is like a triglyceride, convert that using Yerwaria lipolytica into beta carotene or astaxanthin, these nutritional supplements that are pretty expensive. And from a high level, the sort of the workflow is the oil goes through beta oxidation to create lots of intracellular acetyl-CoA that can then go through the mevalonate pathway to make terpene products. So it sounds straightforward, but there's some gaps. And so why did we choose Yerwaria lipolytica? Well, a little bit about it is that it's an oleaginous yeast, and this means that it can accumulate lots of lipids, up to about 40% of its dry cell weight. And the product is sort of a lipid product. It has a high flux mevalonate pathway. So when I say flux, that means like reaction rate. So high flux means high reaction rate. Uh, and the mevalonate pathway converts acetyl-CoA to mevalonate. And then mevalonate can then be converted into all these high value products like cancer medications, jet fuel, perfumes, steroids. So it's a very high value pathway. So the fact that it's natively highly expressed is a very good trait. One key challenge, though, is that the mevalonate pathway requires high amounts of NADPH. So we're very interested in how can we supply enough NADPH to keep the reaction rates fast. And last, even though Yerwaria is not exactly a model species, there are very good genetic tools. So we can pretty easily create new strains. So what are some of the questions that we're interested in? When triglycerol is outside the cell, first lipase enzymes are secreted and break down the triglycerol into the glycerol backbone and then the long-chain fatty acids outside the cell. And then we want to know how these two components, once they enter the cell, are broken down. So we know that I guess we're most interested in the fatty acid side. So it goes through beta oxidation in the peroxisome, and this generates large amounts of acetyl-CoA and NADH. So we want to know how are those two components processed after they're broken down, and how is the NADH converted into NADPH, which is needed for that mevalonate pathway. What we wanted to do was compare the metabolism of Yerwaria lipolytica on glucose, glycerol, and oleic acid. So in this case, oleic acid is a stand-in for, or it is an example of a long-chain fatty acid. And we're using glucose sort of as the control. And glycerol is the backbone component of the triacylglyceride. And we see that glycerol is the preferred carbon source, and it has the fastest growth rate. But then when we look at the yield coefficients, when they're normalized for the number of carbons, like glycerol has 3 carbons, glucose has 6, and oleic acid has 18. So when you normalize, the yields are all about the same per carbon. So the big difference is in substrate uptake rate. So that's why the glycerol has the faster growth rate. Next, we wanted to look at the transcriptomics. So we wanted to know how is the gene regulation different with these 3 carbon sources. And so this first plot is showing differentially expressed genes. So it could be up or down, just differences. And we see that glucose and oleic acid have the most different regulations, the most different number of differentially expressed genes. And glucose and glycerol have the least. And this sort of makes sense because glucose enters the cell at the top of glycolysis, and glycerol enters in the middle, and then oleic acid through the acetyl-CoA sort of enters at the TCA cycle at the bottom of glycolysis. So they sort of match where they enter the metabolism. But then taking a closer look at like which genes are up and down regulated, we made this volcano plot. And so I want to focus on the purple and blue dots that are comparing glucose to oleic acid and glycerol to oleic acid. And we see that in general, it's not a symmetrical plot. Like there's a lot more points to the left side. And this sort of means that when genes are differentially expressed, they tend to have higher expression in oleic acid than those two other carbon sources. And we have two hypotheses about why this might be. The first one is that just more enzymes are needed to break down oleic acid because it's like a more complex carbon source. And the second one is maybe the glucose genes are just always on, and the oleic acid genes get turned on and then turned off. So like the more of a switch there. But either way, there's going to be more metabolic burden when the cell is consuming oleic acid because it needs to produce more transcripts and therefore with more proteins. Okay, the next technique that we use is called 13C metabolic flux analysis. I'm going to try to give a brief overview about how it works. So bear with me. So the way that it works is it's a way to measure intracellular reaction rates. And so you start out with positionally labeled carbon sources. So for this glucose, like carbons 1 and 2 are 13C, the like, heavier isotope. And then the other four are carbon 12, like the standard isotope of carbon. And then when the cell consumes these carbon sources, the labeled carbons don't like go randomly or evenly through the metabolism. So based on the branch points, certain parts of the glucose molecule get like built into certain amino acids based on those the split points of the metabolic network. So we can measure the amino acids labeling patterns, and this is called the mass isotopomer distribution. And we can't tell what position everything's in, but we can say like, okay, this amino acid has like 20% of it has no 13C, like 20% has one 13C, and so on. And then from these mass isotopomer distributions, if we have a metabolic network, we can run simulations of different possible configurations where all the reactions are balanced, and then determine, okay, for this given simulation, a set of reaction rates, what would the mass isotopomer distributions look like? And then it's a matter of trying to find the set of reactions that have the mass isotopomers match the experiment. Okay. But so to do that, we need to have a well-defined metabolic network. So we wanted to remove a couple of reactions in order to have the math work better. So we relied on the transcript data. So for this reaction, like the carnitine shuttle and ATP citrate lyase, for ATP citrate lyase and oleic acid, very low transcripts. So we said, okay, it's probably not active. So we removed that reaction. And same with the carnitine shuttle for glucose. So we removed that reaction. And then this is the result. So it's a little bit overwhelming, but I'll try to walk through it. So this top right here on the left, that's glycolysis. And then here's the TCA cycle in the mitochondria. And then this is like the pentose phosphate pathway. And so every reaction has three numbers. And those three numbers correspond to the reaction rate in glucose, glycerol, and oleic acid. And all the numbers are like normalized to the uptake rates of oleic acid entering the peroxisome at like 0.86 and glycerol over there and glucose. Okay, so we're most interested in the oleic acid because that one hasn't been done before. So the key finding, one of the key findings was that the glyphosate shunt is highly active. So we see sort of like the carbon flows like sort of into the mitochondria and then back out and then around there. Uh, and eventually it flows all the way up to the pentosphosphate pathway. And that's the key place where the majority of the NADPH is generated, which was not exactly what we would have thought. We would have thought that would have happened in the TCA cycle. And last, there's two steps in the TCA cycle that lose a carbon dioxide. And both of those tend to have very low CO2, tend to have low reaction rates. So it seems like the cell is sort of optimizing generating the energy molecules from reactions that don't lose carbon. So it's sort of holding on to the carbon. Okay, so I want to talk a bit more about the glycolate shunt. Uh, so after the oleic acid goes to acetyl-CoA, it can sort of branch into the TCA cycle. But if 100% of the carbon went through the TCA cycle, then it would all be lost as carbon dioxide. So this sort of pathway is a way to get energy, but at the expense of the carbon. And then the glyphosate shunt is you get less energy, but you get to keep the carbon and you can use that to make the sugar. And so we found that is roughly about 20% was used just for energy and 80% was used for uh, just making all of the carbon that the cell needs for growth. And then so we wanted to validate the 13C metabolic flux analysis findings with transcripts. And it's important to note that it's not a one-to-one. Like just because there's high transcripts for a given reaction, that doesn't mean that there's going to be high reaction rate because there can be like regulatory steps, like some sort of like allosteric regulation that would like turn off that enzyme, but they should correlate somewhat. And so just focusing on that mevalonate shunt, um, the glyoxylate shunt, we see that the transcripts for oleic acid are high for those two key reactions. And that sort of matches with the measured reaction rates. And then same with one of the steps in the TCA cycle where the oleic acid has high transcripts and that corresponds to high reaction rate. So we took this as validation. Okay, so far I've been talking about these about 50 reactions of which we call essential carbon metabolism, but there's a lot more. So in order to tell sort of the full story, we need to use a bigger metabolic model. And these are called genome scale models. Um, and so you can sort of think of them like this pipe diagram where the key assumption is that the cell is at steady state, so no metabolite is changing in concentration. So this is true during like exponential growth. Um, and so pretty much all of the reactions that produce a given metabolite have to equal all the reactions that consume it. And they can be used for understanding how the cell will respond to changes in its environment. So like low oxygen or different carbon sources or genetic changes. Like if you were to knock out an enzyme or add an enzyme, how would that affect the like rest of the network? They can be used to calculate theoretical growth rates. And all this combined can be used for computational strain design. So saying like, what is the best set of like gene knockouts to do to increase the yield of my product? But the problem with the really big models is that they're very flexible. There's many different like solutions to solve them. So we want to try to use as much data as we can to constrain them. So uh, what we wanted to do was use the 13C MFA confidence bounds for the different reactions as the bounds in the genome scale model. And then once those 50 reactions are constrained within whatever bounds they have, uh, we can then say, what are all the other reaction bounds? Like what's possible for all those other ones? And that analysis is called flux variability analysis. And this is just sort of a demonstration showing that when you have default bounds, they're very big. And then as you add more and more constraints, they get tighter and tighter. And this is what we saw. So when it was sort of not constrained with the 13 CMFA, there's a lot of reactions that have very large bounds, but then as you add the constraints, all the reactions get sort of pushed to the left, so to a smaller bound size. And just want to quickly note that this is like a log scale. And we see the same thing with oleic acid. So we wanted to use this information to get back to the NADPH source and sinks. And okay, what are the key ones? So we see that in general, a lot of them are like right around 0, so pretty low reaction rates. But just taking a closer look at glycerol and oleic acid, we see that the NADPH typically is primarily from these two pentose phosphate pathway reactions, but also a little bit from the TCA cycle, like I said, about 20%. Um, but there's a lot of questions about the folate cycle, so we still don't really know whether that's a source or a sink. And same with this glutamate dehydrogenase reaction. So I guess whether more like amino acid production or consumption is driving the NADPH production. Yeah, so this work we like applied the first 13 CMFA to a large long chain fatty acid and found the sources and sinks. And it's also a novel integration of the 13 CMFA and genome scale model using the flux variability analysis. And I really believe in publishing all the code for any paper, so that's all on GitHub. The next story also involves Yawaria lipolytica, but this time trying to produce sustainable aviation fuel. So again, we're starting with waste lipids, and then we're going to use Yawaria lipolytica to make like this sort of intermediate product. And then from there, just sort of two somewhat simple chemistry steps can be done to convert that into the 1,4-dimethylcyclooctane, which is a drop-in like aviation fuel. So you can just put it into the tank, or at least mix it. And so again, we're talking about the mevalonate pathway. So we're going to be looking at glycerol, glucose, and lipids going to the mevalonate, going to acetyl-CoA. And then from there, they can go to mevalonate phosphate. And then there's a branch point where uh, mevalonate phosphate can go to the left to like IP and then isoprenyl, or to the right to mevalonate diphosphate and then IPP, and then many steps later to the carotenoids. And so the pathway to the right is a required pathway. Like if you were to knock out like one of those enzymes, the cell was not able to grow. So it makes very important membrane lipids. So you can't completely knock that out, but maybe we can tune the cell so that we're making a lot of product uh, without killing it. And so you want to know like which of these enzymatic steps are the most important or should be focused on for uh, like genetic engineering. So we're going to be using what's called like kinetic modeling. So this is sort of the system where we have 6 different reactions, and they're each going to be modeled as Michaelis-Menten uh, enzymes, but we don't know any of the parameters at this point. So as a quick refresher, the way that Michaelis-Menten works is you have 2 enzymatic parameters, Vmax and the Michaelis-Menten constant Km, and the reaction rate of the like enzyme I is Vmax times the current substrate concentration divided by the Michaelis-Menten constant plus the substrate concentration. And if you're more interested in code, it just sort of looks like this. This is the function, and for the sake of this, Kcat and Vmax are going to be assumed to be the same. Okay. So in order to figure out like which of those reactions is most important or which enzymatic parameter is the most important, we're going to do a lot of simulations and see like which parameter has the biggest effect on the final output. So here's one example set of uh, parameters, so it's generated randomly. And then we can simulate time course data. So for this first simulation, we see that a lot of, or more carotenoids were made than isoprenyl. But on its own, it's hard to say why that happened. So we want to do this a lot of times. So we did it 10,000 times. And these are the first 16 results. Um, and so looking at the outcome of those 10,000 simulations, it seems to be that there's sort of a attractor state where there's a lot of uh, a high probability that the isoprenyl concentration gets maxed out while the carotenoids go to zero. Um, and then there's relatively few in the middle. But we want to know which enzyme is most important. So it's very noisy data, clearly, but we do see so this is the mevalonate phosphate MVAP to IP reaction, uh, also known as phosphomevalonate decarboxylase. And we see that this reaction has the largest slope. So depending on what value you use for kcat, we do see a correlation with the final amount of isoprenyl. And even though it is very noisy, the p-value, I guess, is very small, less than 10 to the minus 228. So converting all of this into an easier to read format, we can say that the most important enzyme in this system is the MVAP to IP reaction. And then the second most important one is the MVAP to MVAPP reaction. And so we want to turn that one down. But that's a required enzyme. So that's a bit of a challenge. And another challenge is the MVAP to IP, the phosphomevalonate decarboxylase reaction, is thought to not have a specific enzyme in Yawaria. We don't know. So we assume that the ERG19, the MVAPP to IPP reaction, that one has promiscuous activity. So it's able to do both reactions because the substrates and products are pretty much the same, just with one less phosphate group. Yeah, so we wanted to use this information to make a genetic engineering strategy, like a strain design. Yeah, pretty much already said that. So this was the approach that we took. So we looked into literature and found that there was only one phosphomevalonate dehydrogenase that had measured kcat and km parameters. So we're not sure if that's the best one, but it's the one that we had to go with. And it's from an archaea species. And then we also added phosphatase from E. coli to do that reaction from IP to isoprenyl and knocked out one of the kinases that we think is doing the IP to IPP sort of side reaction that's taking away from our pathway. And then the ERG8, the mevalonate phosphate to mevalonate diphosphate reaction. So that's the one that is essential. So we can't just directly knock it out. But this strain design was done in collaboration with Dr. Wei-Wen Su or Dr. Winston Su. And his lab, they've been focusing on for the past about 5 years on what they call a de Graan system. And what that is it's an inducible degradation. So essentially you put on a short tag, I forgot how many amino acids, but it's about 5 to 10 amino acids onto any enzyme. And then when you add auxin, a different enzyme will come over and then degrade that enzyme. So essentially what we can do is have the required enzyme be present while the cell is in the exponential growth phase, but then as it enters the production phase, add the auxin, degrade that enzyme, and sort of direct all the flux towards our product. And so here's some preliminary results. So unfortunately, we're only at the milligram scale, but we have been able to show like isoprenyl production. And a big challenge is that isoprenyl is a volatile compound, so it can be like evaporated from the media. So it shows a weird thing where like it decreases in concentration as time goes on. So we've been looking into different strategies for how to capture it, including like a dodecane layer. But this is sort of ongoing engineering work. Okay, so then the third story that I want to tell is about a thing called impact DB. And so the question that this one sorts is aimed to solve is uh, the fact that synthetic biology data is all published. There's lots of data, but it's all published in like Excel files that are supplements, like supplementary data. And there's no standardization. Like everybody just has whatever columns they want, whatever rows they want. Uh, so uh, what a lot of people have done is like they go through and read those papers and then make their own like new data table. Uh, but it's a lot of repeated work. So if there was a centralized effort where everybody works together to make like one sort of well-curated database, that would be good for everybody. So that's the goal of ImpactDB, or the Industrial Microbiology Publication and AI Crowdsourced Toolbox. And it's a website and database. And so yeah, it's impact-database.com. And I'm gonna kind of show you like some screenshots to show how the website works and how it's organized. So it's organized into like genuses. So these are different yeast and bacteria genuses. And then inside of like species or genus, then there's a bunch of papers. So if you click like say on the Yerwaria button, you would then see this sort of like Google search result list where it shows the different papers and the top results. And if you click on like a given paper, you can then see the abstract and a list of fermentation results. And so here's what, after you expand the fermentation result, what it looks like. So it has bioprocess conditions. So that's things like, yeah, the temperature, carbon source, time, and then also the genetics. So which genes are knocked out, overexpressed, or like heterologous, so like from a different species added in. And also the titer, rate, and yield of the product. And it's sort of set up as a machine learning problem where the features are the conditions and genetics, and those can be used to predict the like final outcome of the fermentation. So it's crowdsourced. So there's like a form that users can upload data. But yeah, this is another form users can upload papers. And so you can see that at the beginning, it sort of was growing sort of incrementally. And we were doing a journal club, and we would meet and we would sort of like together work on adding new results. But it's very slow. So then you can see there were some big jumps, and those big jumps came from AI. Yeah, so we wanted to see like how could we use tools like ChatGPT to extract the data sort of for us. And so this was a early approach from like 2023. Um, and these are the exact prompts that we used. So we asked the AI agent to make the multi-column spreadsheet and gave the name of the columns, and then added the abstract and result text. So I guess a key sort of like missing thing from this is the figures. Is like only the text portion was included in this. And the workflow has like constant human supervision. So yeah, we wanted to make sure that there's not just a bunch of repeated numbers or numbers that aren't in the paper itself, and just make sure that it's yeah, high quality data. So what we did was we wanted to just like evaluate how good the AI data was, the AI extracted data. So we had 100 papers that were read by people and 100 papers that were read by the AI. And importantly, those are two separate sets of papers. So it's not a perfect experiment, but it was good for increasing the size of the database. And we see that the feature variance is roughly the same between the two like methods. So we take that as a good sign. Um, and when we look at like a PCA of the data, we see that there's maybe a bit more separation between the different product classes with the AI extracted data, but that the silhouette score and mean distance between the clusters is pretty much the same. So I think this is close enough to uh, like close enough to each other that the data is reliable. And then we use the extracted data to train machine learning models. So these are all like random forest models, and it works very well overall. The places where it has the hardest time are when there's limited data. So like the polyketide data set, like that light blue one, is limited, so the fitting is not so good. Uh, and because it's a website, we can have like interactive tools. So uh, Jeff Chaika was like a fellow grad student of mine, and he made this machine learning model for predicting Yawaria titers. And it actually relies on genome scale modeling to create some of the features. So like glycolysis flux was one of the inputs, and TCA flux was another one. But if somebody wanted to use his model, they would need to like go to GitHub, like clone his repository, set up a Python environment, like run a Jupyter notebook. Just a lot of steps that the maybe average person wouldn't be able to do. So we put the like that model on Impact, and now it's just like a bunch of drop-down menus. And so just much more user-friendly and more accessible. But for the more tech-savvy people, we want to make sure the data is open and like available to anybody that wants to use it. So there's an API. So this can be used for sort of like pulling all the data. Sort of, it's like a way to download the data, but in a way that is easy for other people to write code on top of. So people can write their own models with this data. And some collaborators are working on knowledge graphs. So trying to convert the sort of like spreadsheet type data into a more dynamic, like concept focused format. And so yeah, this sort of graph is what it looks like. So here's a closer zoom in where like more similar concepts or concepts that appear together in the abstracts of papers are linked more closely. And then those knowledge graphs can be used for retrieval augmented generation or RAG. And that can be used as context for these like large language models. So this is comparing Quen, one of like the Chinese open source models, with the knowledge graph compared to just ChatGPT by itself, like without any information. And we see that when you ask for like suggestions for strain engineering and bioprocess conditions, you get a more complete result with the knowledge graph. So we want to make that available to users. So we're working on an LLM page where yeah, people can have a conversation like with ChatGPT, but specifically focused on the bioprocess optimization. And this is a like sort of continuing work. So in the same vein, but this work was applied to cyanobacteria. And so there's a lot of AI extracted information that was then used for techno-economic analysis. And essentially what they found, or what we found was that as you scale up the fermentations, then you get much more profitability. But anybody that's worked with cyanobacteria knows that like a key challenge with scaling up something that's photosynthetic is that once you have high concentrations of cells, they can like occlude to one another or like sort of it blocks the light, so it doesn't grow as well. Um, so going forward, I didn't talk about this, but the sort of thing that I'm the most interested in at this point is like genome language modeling. And so sort of applying LLMs, but that are, instead of them being trained on human text, having them being trained on genome sequences or protein sequences. And that sort of information can be then used for understanding the function and structure and location of enzymes and lots of like regulatory things. So I really think that going forward, understanding how genomes work, we will be using large language models. So try to get into that space. I'm also going to be continuing the Bavaria, Lipolytica, isoprenyl modeling work. Um, and I'm working on this some grant proposals about uh, producing prostinosteroids. So these are plant hormones, and they can really help cells like as they respond to stressful environments like drought or heat stress. But the current way of producing them is like grow the plant, then extract just a very small amount from the whole plant. Uh, but if we can put those same enzymes that make these steroids in the plant into yeast, we can just do a fermentation and create a large amount much more cheaply. Finally, yeah, continuing to grow Impact EB and adding more and more features. Okay, so yeah, I didn't do this work alone. I want to thank everybody that helped me, especially my two current students, Jacob White and Luke May, and thank all the funding sources. Yeah. And if you're looking to get involved, yeah, here's my contact email, my contact info. Yeah, please send me an email. And now I'm ready to take any questions that you might have. Thank you.